Gram-negative bacteria are surrounded by two membranes. There are lots of porin proteins in outer membrane of this bacteria. Each porin forms an open water-filled channel that allows passive diffusion of nutrient and waste products. These channels are restricted in size, and this excludes larger, potentially toxic compounds from entering the cell. Porin protein folds into a 16 antiparallel beta barrel. This protein contains a central channel because of a large number of beta strands. The channel is partially blocked by a long loop region between beta strands 5 and 6, red one. This arrangement creates a narrow region in the middle of channel called islet, about 9 angstrom long and 8 angstrom in diameter, which defines the size of solid molecules that can pass through this channel. One side of this islet channel is lined almost with positive charge of 1 histidine, 2 lysine, and 3 arginine. The opposite side of the wall with negatively charged residues, 4 glutamic acid, 7 aspartic acid. The large local surplus of negative charges is partially compensated by 2 bound calcium atoms. This asymmetric arrangement of the charges, no doubt, contributes to the selection of molecules that can pass through this channel. Bottom loops are very short, making flat and smooth surface facing periplasmic space. 3 molecules of such a barrel assembled to make a trimer. You are looking at the trimeric structure of porin viewed from the extracellular space. Blue regions indicate the wall of the 3 porin barrels. Loop regions that constrict the channels are red, and the calcium atoms are orangey. Large molecules are prevented from entering by the size of funnel of a trimer, this one. Further screening occurs at the entrance of a channel in the individual barrels. Finally, a molecule small enough to enter the central channel then encounters the islet, where the charged side chains determine the size limitations and the ion selectivity of pores. This protein is affected by the environment, the conditions. Changes in an electric potential or osmotic pressure can modify the structures of channel and therefore its permeability. But as yet, we have no structural information on such changes. Next, membrane protein will be about potassium channel. There are diverse types of ion-specific channels in membranes, such as sodium, potassium, calcium, and chloride. All living cells show membrane potentials that just made by sodium-potassium pump. In addition, the potassium channel just leaks potassium out of cells. You can see the schematic diagram of the structure of a potassium channel viewed perpendicular to the plane of the membrane. The molecule is tetrametric with a hole in the middle that forms ion pore, the purple color. Each subunit forms two transmembrane helices, inner and outer helix. The pore helix and loop regions, this one, build up the ion pore together with inner helix. This diagram shows side view of two subunits of the potassium channel, illustrating the way the selectivity filter is formed. This one in ion pore region. Some loop connecting pore helix and inner helix forms selectivity filter. Main chain atoms of this loop line the walls of this narrow passage with carbonyl oxygen atoms pointing into the pore. The tight packing in this region allows well-fixed alignment of carbonyl in selectivity filter, just fixing the diameter in pretty much fixed size. We must explain the feature of potassium channel based on the molecular structure of this protein. Potassium channels are highly selective for potassium ions by a factor of 10,000 over sodium. What is the chemical basis for this selectivity, since both potassium and sodium are just a spherical shape? Simple size exclusion cannot account for the selectivity because sodium is much smaller than potassium. Something like this. Usually, high selectivity implies there is a strong interactions between potassium ion and the pores, so that the release of bound potassium from pores may slow down the passage of potassium. However, this channel has a high passage rate close to the diffusional rate of potassium movement. The diameter of ion pore varies along its length, wider and much wider at the middle, and narrow. Cytosolic half of this ion pore is filled with water. The negative charges near the filter, such as C-terminal alpha helices with a negative dipole moment, providing negative charges in this region near the selective filter, attract potassium ions, which is positively charged. In addition, two prominent features of selective filters could explain further. First, the selectivity filter has three closely spaced binding site for coordinated naked dehydrated potassium ions. The coordinated property of this one could imply that potassium does have only a small distance to diffuse from one side to the next binding site. Second, tight binding of side chains of selective filter with neighboring pore helices packing firmly, fixing alignment of carbonyl group in selectivity filter. Based on these things, altogether, selectivity could be explained by dehydration energy compensated by binding of carbonyl group in the selectivity filter. The high rate could be explained by combination of three things: coordinated binding and movement of potassium ion in the selectivity filter, and also charge repulsion by closely located potassium ions in selectivity filter. Lastly, potassium ion gradient formed in living cells by sodium-potassium ions. This slide explains again the mechanism step by step. First, the selectivity could be explained by this procedure. Initially, potassium up until middle region has a hydrated water, but when potassium ions binds to the selectivity filter in this region, potassium ion must be dehydrated. This step requires energy, but such energy could be provided by binding to carbonyl group in selectivity filter, because the binding of this carbonyl group to potassium ions exactly mimicking the hydrated conditions in terms of energy. High passage rate are three things, as I mentioned, and there are two types of potassium channels among organisms. Tetramers with two alpha helices for the transmembrane domains, and then another type with 6 alpha helices. But it is highly conserved for the pore helix and the inner helix in this channel, strongly supporting the mechanisms we explain in here.